I just
All right, coming down some more. Greg, would you mind coming down a little bit on the winch there?
Yep.
So, Alexis, do you have plans to look back at the
What's the floaty
video
thing?
to kind of
It's a
study
fish.
amount of different species and distribution, and how do you plan on doing that?
Yeah, so Steve does that with some undergrads in our lab. So
Okay.
they go back through the video, and that's why we try and make sure to do zooms on at least a couple of what we think are the same types of coral, so they
Right.
can make more solid IDs
Right.
based off of the video.
Static.
But
You're gonna go ahead and
essentially,
bring
like,
that around.
they just take screenshots of all the corals and they
Right.
make like a big, like, spreadsheet about it, and then
Right.
they can go back and reference with each other and trying to figure out how many different morphospecies
Yeah,
there
go into
are,
my
so how many
pod.
different morphologies, and then they essentially like count them across depth, and he will plot them across the depth range to look at their distribution, and then across the different seamounts as well, essentially, to
Okay.
try and determine if there's patterns of like similar depth ranges, or if there's patterns like based on water masses
Okay.
and things like that.
Okay.
Yeah, so they use all this. I mean, we use all of this video data. It just gets rewatched much more closely, and we
Right.
really confirm the IDs.
Right.
I mean, it just, so what you're describing really is one of the things that makes this type of work so important, and just underscores the fact that we have these massive areas that we're trying to protect and manage, and we don't really have that basic information of like, all right, what's there?
Right.
Like, what are the species that are there, and what's their distribution? Where do we find them? What is a healthy baseline look for these places? And that's what these types of research trips really can
Yeah.
help build that information.
Yeah. And like this site being a lot more sedimented is something that and by sedimented we're seeing a lot more sedimentation, not only on the rocks, but we've seen it on some of the corals, and we've seen it on the sponges as well.
Yeah.
And that can explain some of the like maybe lack of coral abundance here, and using that information when doing these distribution studies as well is really important.
With so much that we are still yet to explore,
Marley,
how do we
location there,
how do we
please.
extrapolate this distribution to?
Oh, there's a chimera. That's a chimera to the right.
Oh, to the bottom right there.
Oh.
Sorry, go ahead, Ben.
how do we extrapolate this to kind of better describe the entire monument?
Yeah, so we use presence absence data essentially. So like presence is showing that there is a certain type of coral there, and absence is saying that it's not there, to input that data into models, which can help predict where we might find similar assemblages of corals. And so the more points you have in your model, obviously, the better that model becomes. And so we can start to, this is like, and this is baseline data, like we've said. So we are just starting to create the points to go into models for the Pacific. The Hawaiian Islands are a lot more vastly studied, so that's really great. There's information up there, but down here, around these remote Pacific islands. There's still just a lot we need to gain from doing these types of surveys.
Well, and just for some scale, so if you leave Honolulu, it takes about 5 days by ship to get to the end of the Northwest Hawaiian Islands. So that encompasses the entirety of Papahanaumokuakea Marine National Monument, whereas it takes 7 days by boat from Honolulu just to get to Howland. And
Oh,
Baker.
longer, yeah.
Yeah.
Yeah, 7 is about
Like
right.
if we're
Yeah.
not doing any other stops.
Right,
So
it's
if we're
about
just like
7.
trucking by boat, that's how long it takes just to get
Yes, yeah.
So you need a faster boat.
here. Yeah. We're gonna need a bigger boat.
It's true.
You give me problems, I will solve them. It's not going to be helpful, but it'll be solved.
Faster boat's very helpful.
Yeah.
Speed Nautilus.
Extra
Turbo.
zoom.
Yeah, well, we had like, what, 13?
Lasers 12.
Lasers to 12.
Is this a shadow here, or is that a large black coral?
Where?
And right in front of the lasers, it looks like a shadow now that I'm getting a little closer
Oh, I
to
see.
it.
Shadow.
Yeah.
All right.
Looks like a solitary tentacle.
I forget where it was, but we were following a fissure with the lasers, right?
Oh.
So the lasers were right in this fissure that was probably about, I don't know, maybe 20 centimeters wide.
Huh.
So that was the running joke, was we had the lasers on blasting power.
Creating the vision.
It was 3 a.m. It was hilarious. Who's on that watch?
What would happen if you set lasers to stun?
Well, you'd stun things, obviously.
But would you stun the rock?
The rock is already stunned, that's why it's not moving. That's how good the
Oh.
stun effect works on the rocks. I mean, you haven't seen a rock move that we shined lasers on, have you?
Hopefully not.
Yeah, well, if not, then we'd have to fix the stun setting.
It's true. We might have other things to fix at that point too, if we see all the rocks start to move.
Like
Yeah,
our
your
sleep,
brain.
our sleep patterns. Yeah.
Your brain.
Boy, we went two totally different directions. I'm thinking like massive landslide
Yeah.
burying the
That's what I was
vehicle, you know?
thinking. It was like time to go.
No,
It's
that was
like,
very-
I haven't slept in 2 weeks.
Psychological.
There is no rock, Ben.
There is no rock.
Look at that. It's
We're
like
actually on
a
the flat
beach
sandy
down
bottom.
there.
What is
Yeah, now
rock?
we're getting at the end. This feature
Well,
looks
you would
cool to
know
me.
more than anyone.
Hmm?
This little
We need a
local
chaise
minimum.
lounge and
That's the only way I can
minimum.
refer
you
to
know,
it
a
as.
coconut drink.
That sounds nice.
Oh, there's a coral over there. See it?
We're just a little outside of scope right now.
Gotcha.
Unless, would you
I
be
think-
able to come down a little
Yep.
bit there, Greg?
I think you can get there. I have faith in your abilities.
Faith in the pole.
Yeah, it's gonna be pretty flat for a while.
Yeah.
Yeah.
It must steepen drastically towards the end.
Steepen.
Yeah, I think it's pretty consistently sloped after this part. Oh,
I love
I mean,
these.
Yeah,
speaking
that's
of current,
towards the-
I know,
mean,
look at
that
the
soft
sand,
sediment,
yeah.
like sort of like channeling out around the rock.
All right, video, go ahead and push
It's
on in there,
really
please.
cool.
It's really beautiful.
Mm-hmm.
You can see it nicely in Argus.
Oh, yeah.
See if I can land here.
Wow.
Yeah, I'm all stable
Thanks,
now.
Megan.
What?
I said thanks.
Oh.
Complimenting my Argus piloting.
Oh,
Yes.
of course.
She's like, yeah, that's what I was doing.
Yeah, good work, Greg.
Yeah.
Great
Don't
numbers.
forget your numbers.
I did my numbers.
Always eat your vitamins.
Yeah. Always take your numbers.
Always take your numbers.
You have any more zoom on there, Marley?
Yeah, yeah. Are you good?
Yeah, I'm against the rock right now. Just getting a little bit blown around, but it's good.
I think this is a Calyptrophora. Which way are the polyps going, up or down?
Looks like down.
Down.
Some of them are both, though. Do you see some of the branches that have...
Yeah.
Depends on the
Is
day.
it possible that it's 2 corals?
Oh,
I
wow.
don't think so. I think it's 1, and I think just sometimes the polyp orientation is confused.
It's a Calyptophora.
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 Jess, you want to peek at the base?
Yeah, that'd be great.
Yeah. I'm just gonna bump down on the camera here.
Yeah.
Do you need me to zoom out at all?
No, it's okay. You just might. I'll close your eyes for a minute because
No,
it's a
look at
video
that.
sin.
This is a nice branch.
Yeah.
Looks pretty healthy, yeah.
Yeah, it does. The tips, there was some hydroids, but for the most part, it's fully covered in polyps, which is good.
It's kind of like swooping down the rock, and we can see there's a pretty good current with the ripples, so
Yeah.
it must be a good feeding area.
Yeah.
Do you want me to hold position? Or
No, that's
no,
good.
we're
We're
just
gonna move
checking
on.
out.
All right, Jess, I'm
You
passing.
ready?
Yeah, we can come out if you...
Alrighty, Alexis. Marley, go ahead and
Yeah,
come
I
right.
got you.
You can see a couple other...
Wow. You can see it like kind of dipping down into that current.
That is really beautiful.
That's awesome.
I love the way, like, there's like the thicker branch and then all the little branches coming off of it. How they're all like in the same direction.
Yeah. It's like draped.
Yeah. It's really beautiful.
I wonder what the texture would feel like if you were to epoxy that and run your fingers over it. Sorry, that's the only thing going through my mind right now is how does that feel?
Yeah. No.
Stroke the
sand?
coral.
Well,
The sand or the
the sand,
coral?
I
The
think.
sand, yeah.
The sand,
You'd
yeah.
have to shrink it a little bit.
Yeah.
We need
At,
those
at this size, it would be a lot of epoxy.
That's
Vacuum
true.
seal it first. Yeah.
lasers
thought it-
to shrink.
I think it would be so cool to have like floors that had like weird rippled patterns, but they were black
Oh,
and they were smooth to walk on. So like just below, kind of like a clear, smooth epoxy, and then below
I
having
love how
like,
you had to indicate
I mean,
the color as
y'all,
well.
like, this
You want a
exists.
floor that's
This is called
full with
sandstone.
salt?
So if you go to the southwest, you go take a look at like different layers of sandstone in the
But can you have it in
southwest.
your house?
Yeah. Yeah,
Yeah?
oh, absolutely.
Oh.
Yeah, I mean, it depends on where you're collecting it, but definitely, like, you can have, like, floors that are, like, flagstone made out of different types of sandstone that have these ripple patterns in them.
Wow.
That are
Cool.
literally, like, just naturally epoxied.
Amazing.
It's super cool.
I've never seen such floors.
Oh, it's pretty great.
Our science enthusiast is just so
Also
enthusiastic.
architecture.
I was going to say,
enthusiast.
Megan, you should travel, but I feel like
I was thinking you should travel. Except,
Just floating around the middle of the Pacific.
You are well traveled.
I've never been to the like southwest of the U.S. though. Actually,
You would dig it as a, volcano
I
enthusiast.
I know I would.
You
I
really
know.
would.
I know.
mean, because you get everything. You'd get like your ripple rocks. You'd get volcanoes. There's so many volcanoes in the Southwest.
I know. I'm dying to go.
Mm,
It has to happen.
do it.
that's a type of fish is that?
oh, yeah, who's that? Oh, sea pin. That's a sea pin. And then
pin.
what's
Hi,
in
sea
front of
pin.
us?
I think that's
Yeah.
another.
Rat tail?
No.
Yeah. I was
Looks
hoping
like
for
it.
tripod
Oh,
fish.
yeah.
Yeah,
I'm
I
seeing
remember that
I'm
as-
seeing
You
the
gonna do a
shadows.
snap zoom in there?
Oh, the
Marley?
shadows,
I thought it
In
had
an
yeah.
environment
multiple
just like
legs, it was
this,
just
they
a
were
shadow.
all
Marley,
like
go ahead
just
and zoom
sitting
on in
here.
there.
It has multiple, like, fins, right?
It does, but I thought it was like
Yeah.
So
multiple
fast
of the,
does not
the
want us to
undulations.
do
That's
Okay.
different than the ones we've been seeing,
Yeah.
Yeah.
though.
Yeah, I think it's like the pectoral and
They're
the
really
pelvic
trucking
fins.
too.
Yeah.
Yeah.
The dorsal spine is crazy. All very spiny.
All right.
All right, getting off that course. Okay, let's go ahead and come wide
Off
there.
in the rhubarb.
Off in in the rhubarb on sandy flats.
Rhubarb.
Oh, there's
I
something
love
ahead,
rhubarb.
too.
Little sea pen.
Oh, that's another.
Forget if it was Mike or Trevor who I heard say that first, but I was like,
sea pen
that's
with
when
their
to keep.
peduncle.
That's a good one.
What?
Buried in the sand.
Mm-hmm.
I wonder what these little black blobs are.
Did you want me to look at those?
We might do just a sediment zoom.
Yeah. Want me to push ahead and then
Yeah,
sit
put-
down
yeah,
and do
yeah.
a sediment zoom?
Sure.
That'd be kind of fun. Never done a sediment zoom before.
Really?
Zooming on sand.
Yeah.
Sand.
People usually don't want to see sand and have it like a enthusiastic, yes, let's see some sand.
I know.
So I dig it. Let's do it. I'll zoom ahead and we'll have as much time to the sand as we want. How about that?
Oh my gosh. I only need like 12 seconds and then I'm over sand.
I feel like Herc's shadow right now in
Ooh.
Argus kind of looks
Batman.
like our no, it looks like our little kayak
yeah,
fish, right?
Yeah.
Needs more hits.
Ooh, look at that. Nice.
That's like a bat signal.
That's
No,
totally.
yeah.
Very much a bad signal.
Or like a manta ray.
Ooh,
like
yeah.
Robotic manta rays.
That'd be a sweet robot.
I
Right.
like this.
I mean, we have a
Alexis,
sweet
you
robot.
wanna
I mean, yeah, we
anywhere
just-
around here?
we have a couple of sweet robots.
I'm gonna do a small little stamp.
Yeah, that's a good spot because I want to zoom on that dark little black spot to the left.
Dark black spot to the left. Roger. You'll
One
just have
that's
to
like
show
Oh,
me.
and
left
there's
of
a
the
coral.
lasers halfway off the screen. It
Yeah.
looks like there's a whip coral off in the
Yeah,
distance.
there is.
How about I do a little rotation? The dark spot that's kind of almost off screen.
Yeah.
Something like. Let's go a little further to it then.
Yeah, you're close enough to get a good zoom on it.
All right, Marley, go ahead and push on in there.
All right, are you guys ready for this? This
Yeah.
is going to be epic.
What is it?
Journey through space.
Dun, dun, dun. Do, 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 do. This feels like that Instagram filter where it's zooming in.
What are you?
Is it a little
I
holothurian?
think it might be, yeah. Oh, and there is one right in the center of our screen.
Yeah.
Yeah, see him? Little
Look, it
clear
looks like
one.
he's-
Oh yeah.
Oh, there's a couple of
there's
them.
two.
Yeah,
Yeah,
it's
one
at
on
least
the
two.
side.
They're having a
I
party.
bet they're
Look at the
all
one on
over.
top.
Yeah, the
right.
little red
Yeah.
one, I think is also a holothurian.
Is it
Just
are you getting,
different?
like some motion?
Yeah.
on it right now. Yeah.
Let me know if you want me to look around anymore on the HD cam.
Yeah, we can just like kind of pan up,
Oh.
see, or just kind of pan around, see if there's, or what else is.
Not much.
What are
over
those little
here.
white bits?
I don't know for sure. Maybe pteropod shells.
Pteratog.
Another little holothurian?
Yeah.
Oh.
Sorry.
They're all over, those little
Yeah.
tiny
all
ones.
over. You have any more zoom there, Marley? And then
now
I'll
we're all the
wake
way
up.
in.
Okay.
So if we're fully
Go ahead and
zoomed
come
in,
on,
that's like an inch if
Oh,
not.
if that, yeah.
Sorry,
Yeah.
what was that, Ben?
I just put in another set.
Sure.
Do we like sample
come right
sand
there,
like
Marley.
this at all?
Sometimes it
Sometimes.
depends on who the scientist is.
If anyone's looked for it?
All right, let's push back out and get in a nice
Yeah.
configuration for the other group,
Other folks, yeah.
those guys.
Those cute little red holothurians.
Yeah. That's pretty neat.
The clear ones are weird.
Yeah, and I've seen those in the really similar ones in the Atlantic as well. I think they just are honestly everywhere, and they just like process sediment like crazy.
Nice plume leaving behind.
Bird sounds out there. Yeah,
Oh.
it's crazy sounding.
Oh.
Those birds?
Yeah, it's like, what is
That's the city turns. So we should be, we just opened the door to the van and you could hear like this cacophony So those would be the city tern colony. They are really active at night, and there are about 1 million breeding pairs on
my
Baker
gosh.
Island.
What?
Well,
I'm
when
excited.
they're all there, so like I'm not gonna. They're
Yeah,
probably
yeah,
not
yeah,
all
yeah.
there right now, but like
Yeah.
So but city, it's gonna be loud night on the deck.
Wow.
Bird colonies
Cacophony. I like that.
are really loud. Like you have to wear ear protection when you work in a bird colony.
Oh,
Is
really?
that true? Wow.
You should be wearing ear protection. Like, our field biologists right now are being like, I don't know. Yeah, you should definitely be having ear protection if you're there for a long time.
Wow.
Like cumulative power.
Hey.
You hear the cities? Yeah.
That's so
Yeah.
wild. Yeah.
I'm gonna when we're out of the van.
Yeah,
I'm
you'll be
gonna
out of
go
the mud
take some
pretty
video
soon.
for Beth, for our wildlife biologist.
Yeah, we're on the edge of this thing, so it depends if.
All right. Plenty ahead now.
Hey.
Good morning.
Are we stepping or tracking?
I am just doing 40-meter steps, but we can track a line if you want.
No, that's just fine. I'm
Alright.
happy
Set
to.
1, set 2, set
happy to
3,
not.
record alpha.
All right, I'm
record
gonna
do
Bravo,
another,
check,
yeah.
check, 1, 2.
Ben,
Mic check, 1, 2. 3 green, 3 green.
could you please
9,
bring up,
30.
Nav, could you bring up the backscatter?
Sure, yeah.
Three.
It's going to start.
Or is it on, let's see, I haven't checked high pack plan.
Yeah, so it's all loaded up
Oh,
here.
there we go.
So you can kind of select it.
I see.
So the thing is, sorry, we're going to lose it up toward the north.
That's okay. So we're right near waypoint 2, or 3.
Yeah, we're heading to the end.
Hey, could you come full super
And we're
extra
near
wide,
that
please?
waypoint
Yeah.
3, which was on that
That's
local map.
deluxe wide on both cameras.
So,
Yeah, this is it.
looks like if we
Okay, you can give it past a little micro bump on
just
both.
bump in.
Do like a 060 maybe. We could
Doop.
stay on some
Great,
higher
thanks.
backscatter
You betcha.
and then correct over towards the end.
All right, let me adjust the ship move for that then.
Yeah.
Okay. I see you're really taking advantage of the backscatter data.
It's been pretty reliable.
Nice.
And dive. Cancel. Copy.
So is this, or not?
Not yet.
1784.
No, I mean, not is it currently, but should it be? Or is it just wrong? Is it just wrong to do that? Or is that what we should be doing?
Turn that down. Delete that. Turn that on. Grab that.
Hello everyone, thank you for joining us here on Nautilus Live. This is the 8 to 12 watch.
And we're continuing our way up the slope of Baker Island, the southern slope, with about 2 kilometers left in our track up the slope, and we should be able to complete that by the midnight to 4 watch after ours. 4 o'clock local.
And we're staying on today's date tomorrow, correct?
Maybe we get another September 1st. Yep.
Yeah, bonus.
Which I'm kind of glad that we're finally back on
Yeah.
GMT
We
minus
go from-
11.
I've heard we don't get 2 ice cream days though. That's the rumor though. So I'm hoping for another Sunday, another ice cream day, but we'll
I
see.
think I had enough for 2 days today. So
I should have done that.
yeah.
Yeah.
Oh yeah, we've got a flock of terns right above this ship tonight. We looking west to or north to Baker Island.
That's a long fish.
Through binoculars, you can see thousands of seabirds in flight above the island. They seem to be just staying above the island,
It's
which
crazy out there.
looks like a thin, just a thin sliver of bright coral sand in the distance.
Is that a lighthouse on the
It might be.
island?
Yeah, I think it might be an old lighthouse.
Could you center me up, please? And you can come down on Delta too.
So as everyone's settling, we'll start our round of introductions for those people that are just joining us.
Perfect, thanks.
My name is Tashira Cruz. I'm a Science Communication Fellow here on the Nautilus.
Can hear it when-
My background is in coastal marine biology, and I'm currently an environmental and science communicator.
I'm Emil Petrancio, lead scientist, co-lead scientist with Alexis Wenning for this expedition. Sitting in the chief scientist position back here in the back row. Physical oceanographer by training, and in my 7th season with the Nautilus.
I'm Gina Seelig. I'm an ocean science intern here on the Nautilus for this expedition, and my background is in marine science. I'm currently a senior at the University of Hawaii at Hilo, and yeah, super excited to be here.
Let's hear from Argus. So talkative, Avery.
I'm sorry, what was that?
Yeah, it's hard to get a word in edgewise with you. You're up. A little background.
Oh, hi. I guess I'm Avery. I got my bachelor's of science from University of Texas in Dallas in software engineering, and right now I'm working on my master's in computer engineering from Colorado Tech. Yeah.
Thank you.
Trevor, Herc. No more. I'm Canadian. This is my 4th year here.
Yeah, so what did you work out with using hockey pucks with Herc? What you incorporated them into the...
Hockey pucks are readily available, affordable, and easy to machine raw material. Great for working with the manip. Great for stuff around the house. You know, just very versatile raw material.
On the ice too.
I heard that. Yeah, I heard that you could use them for sports too.
It's a cool sponge.
Yeah, it is nice.
Yeah, well, I had a lot of fun going after pucks that went through the holes in the creek behind my house each winter. Send the lightest kid on his belly towards the...
Yikes.
Can I get the comp and thruster for Argus?
I'm Renato Kane. I'm the navigator on this watch.
So once you get used to that, try to be able to do it from just 5.
Okay.
But if you still need this to reference it, then no big deal.
Every time I'd come out to be navigator, you know, once or twice a season, Rennie would always retrain me because it's enough time for me to forget everything.
No, you were good at all. Although I do like you in this role.
This is fun.
Great chief side.
Yeah. So this backscatter, in addition to using the bathymetry data that we obtained from our multi-beam mapping before the dive, the multi-beam also provides us backscatter information, and it's been pretty reliable so far on this dive. As Rennie was saying last watch, it is relative, but we've
been having luck. It's been good.
Yeah. So the high backscatter shows the hard substrate, which is what the corals and sponges like to adhere to, and so that's where we're focusing our efforts.
Do you get the backscatter from multi-beam or from sub-bottom profiler? Like
from multi-beam.
Oh,
yeah.
wow.
So
Okay.
it's the intensity of the return of each individual ping.
That's different than the sub-bottom profiler, correct?
Yes, the sub-bottom is a single beam sonar, and it is a low enough frequency that it penetrates the seafloor.
Okay.
And we get returns of different densities down to, you know, depends on a lot of things. Sometimes we can, obviously, if it's basalt, we're not going to penetrate that and get layers underneath.
Sure.
But if there's some basalt and then you have like a valley of sediment and you can see different sediment layers, we can pick that up in the sub-bottom.
Are there such thing as multi-beam sub-bottom profilers?
I haven't heard of that.
You could have an array of no, I don't think that I'd have to think about I think that theoretically could work, but it might be practically not practical.
Might be tricky to process.
Because they don't use a fixed frequency, right? They have the
sweep. No, it's actually
Yeah.
it's a fixed one for the chirp.
But doesn't the chirp go like tweet?
No, it's actually for the single beam. No, it's just one single chirp. The multi-beam does have slightly different frequencies, but it's
just so it knows the difference, right?
Yeah, for different sectors.
Sure. Okay.
So the multi-beam was classified technology available only to the U.S. Navy back in the 60s.
Yeah.
And it was actually, the idea was...
It's a brilliant idea.
It was
incredible.
Yeah, it was devised actually for airborne radar, but there was a halt in U-2 flights after
One more step.
the Gary Powers incident when a U-2 was shot down over the Soviet Union. So the developers of the technology went over to the Navy, said, hey, we got something you might be interested in.
Wait, so the Gary Powers plane had that on it?
No, but that was
Shiny.
a U-2, and that was the program that the technology was going to be used for. So since that came to a halt for a while, they went down, put it on sonar, and then it became commercially available, I guess, in the 80s, maybe.
Now, what about the air cannons that are used to get an idea of the seafloor? How does that work?
Those are very loud.
Yeah, I would think so.
And they typically are in an array, I think.
Yep, towed array.
A towed array, but I don't think that they work in a multi-beam capacity. I don't think it's a multi-beam array.
How do they get information from sending a big blast of air down, or a shock wave?
I don't know.
Well, it's an acoustic signal.
Yeah, I'm not sure how. I've never used that technology, so I don't know the inner workings. But
Should have asked Ethan when he was here.
Maybe that's just the way it generates the acoustic ping.
Could be.
Yeah.
Yeah.
Instead of electrically, it
is
is
yeah.
there. This is so much better now that we glued it.
Oh, is it?
Yeah.
Can I see it?
Yeah. It feels like it's going to slip, but it doesn't at all.
Oh, that's good.
Say again? Yeah. E6000.
Now, Emil, are we looking to sample any nodules during this watch?
Any what?
Nodules?
Oh,
So
manganese.
sediments,
If we
nodules?
saw manganese nodules, I definitely want to sample one. But it's more likely that we're just going to see these encrusted, the iron manganese crusts on the basalt here. The manganese nodules, they have been found as shallow as 2,400 meters on top of some of these geos, I think, around Johnston Atoll. But they are normally, I think, more often found on in the abyssal plain, down around 4,000 or 5,000 meters. But yeah, if we come across what look like manganese nodules, I'd certainly want to sample them.
We do want to sample them?
Yes.
Okay.
Cool.
This kind of just looks like broken up basalt. But they're potato sized and
potato salad.
Yeah. Now you might break your tooth on that salad.
So it's like when I cook.
Manganese nodules can form inside, beneath the surface of the sediment, as well as on the top of the sediment. Different formation rates, but
what did you say the rate was?
Yeah, so 1 to 10 millimeters per million years.
That's wild.
Yeah, and then I think the ones that form in the sediment, I might have it backwards, but are a bit faster, 10 to 100 millimeters per million years.
Wow.
Still pretty slow.
So does it take the manganese out of the water?
Yeah,
and
the
it just sticks to it?
manganese. Manganese I think comes out of hydrothermal vents mostly, but yeah, it's in the seawater, and it becomes oxidized. You get manganese hydroxides and iron hydroxides and.
And why does it stick in nodules? What makes the manganese come out of the water and stick in one spot?
Yeah,
and, and also why here versus 100K to the west?
I think we need a chemical oceanographer for that question.
Yeah. Well, what they like is a nice steady abyssal current, and I think let's go check out that nice coral.
All right.
So you get that nice flow up in the Clarion-Clipperton zone. It seems to be a pretty good spot for them, but this is a favorable area. Maybe a low sedimentation rate.
Emily, you wanted me to get a zoom on this one?
Sure. Let's see what that associate is.
Okay, Ed, go ahead. Is that an anemone?
It kind of looks like one.
It looks like it's eating the polyps anyway.
Yeah.
Did I see a nudibranch to the right? What is that?
You mean on the sediment floor, on the floor?
Yeah, what is that?
Oh,
Brian?
that. That's interesting.
Yeah.
Gonna go look at that?
Yeah, we'll take a look at that.
Okay.
That is a nice, interesting color.
I don't know if it's a nudibranch.
Bump.
Just, bridge nav.
Okay, Ed, go ahead.
One more step, please. Thank you.
Ugh. Bump. What is it?
I'm stumped.
Ctenophore?
All right, I think a jelly would be in the water column, not.
Oh, it looks like the urchin sacs.
Yeah.
Those
maybe.
venomous sacs
Oh,
on top
those
of the urchin.
poison, I think. Huh? But
They look a little
Poison or
lighter
whatever.
in color, I think.
Maybe
Could
another
be hard to
angle of
say.
this thing. I can't tell.
I'm wondering if
Yeah.
it's worth a slurp.
Yeah, decide fast, but we
Yeah,
can do it if you
gotta
want.
move on, huh?
I can cancel the move.
Yeah, let's hold here for a sec.
Okay.
Oh. Bridge nav.
All right, come full super wide, Ed.
Ginormous
Thank you.
wide.
Gigundus wide.
Able to
Thank
find anything on this?
What's our slurp jar situation?
We have slurps 3 through 7 open.
That's an
Okay.
array of slurps.
Slurp array.
Let's,
You want me to poke it first,
5.
just see what it's doing?
let's get a zoom.
Go ahead, Ed.
Yeah.
Yeah, stand by.
I think we're flat enough that even if Argus goes over, we'll be all right.
Sure.
You know, we do have some folks interested in the invertebrates.
Can I see preset 4, please?
Huh,
Oh yeah, it seems to be partially open on the bottom.
partially. Yeah.
Would you like me to kind of nuzzle
yeah,
it?
let's just kind of give it a touch, see how it responds. Oh, maybe
Okay,
not
it's opening.
necessary. What do we got here?
Wow, what are you? We might have to watch
Okay,
this.
could you do
behavior.
preset 1
Oh,
again?
it's got little tentacles
Let
on
me
the
come out to
little tube feet, maybe.
you.
The shadow was
Man, that's a
ominous.
scary
I'm wondering
shadow,
if
yeah.
a scoop is. You think a scoop's better than a slurp or a?
Sorry,
We could
I'm
do a
bit
super
tight.
low setting.
You can keep going, Ed.
You
You
sure?
can go all the way, yeah.
All right, there's Max.
It's opening.
That's very tiny.
We could do a really low percentage slurp, and that may help Emil.
Uh-huh.
Yeah, it seems attached.
Yeah.
What are
I'm
you?
being real gentle. I could be a little more forceful.
See if it's attached to a substrate or if it's just on the sand.
Oh. Wow.
Wow, what are you?
Maybe not.
This is interesting.
Yeah.
Egg sac?
Could be some sort of egg.
Is it a weird?
But it seemed to be actively opening
Right.
before.
Yeah. I don't think it's an egg sac. I think it's
Yeah, I
something.
think somebody on the data
I think it
team's
might
gonna
be a sea
patient zero.
cucumber.
You think it's a
cucumber.
holotherian? Do you,
We have
do you
a
want
vote?
this as a sample? Is that Alexis?
What do you think, Alexis?
Yeah, you could slurp it.
Slurp, okay. Let's do it.
It's a Dumbo cucumber.
Could be a gastropod.
Yeah, that's why I was wondering if it was a nudibranch.
Don't know. Gastropod, possibly. Yeah.
Do you get enough images of that to be happy back there?
Could you rotate
Yeah,
me
I
to
got quite a few.
jar 3, please?
Okay.
Now I'm trying to go back into like gastropod characteristics. Don't gastropods all have like eyes and a noticeable mouth structure? Or at least the ones that I've seen before.
Sometimes. This one reminds me of the purple orb we saw.
That was a pluribranch, I think.
If they thought it was a pluribranch,
Yeah.
it's actually maybe a velatunid snail, so a different marine snail with a remnant shell inside.
Hi, Sam.
Hi.
Hey.
Like to
Sam's
just
here.
jump in at key moments.
Yeah.
Okay, could you give me 30% slurp, please? This is going into jar 3. Dunk. Let's keep an eye on it and get ready to 0. You can come wide
a
at
little bit of flow. Wide eye.
Try to keep it as gentle in the flow as possible. As long as it makes it through.
I see a little more flow.
There
now.
it is.
There it is.
You can 0 it and
Ooh.
set it to flush, please.
Looks like a little bat.
Yeah.
Sea bat.
What's the sample number?
Oh, it is swimming around.
That's 30.
Whoop.
030.
There's
Got
flush.
it. Our depth is before. Don't move. I'll do another reset. There,
Roger,
Trevor.
reset.
And Roger, reset. Let me think
Cool.
we're here.
Um,
Are you okay for a reset?
I'm okay for a reset, yep.
yeah, definitely
We are
gastropotty.
reset. Gastropotty. Gastropotty mouth.
Gastropotty mouth. Oh my gosh. Rennie.
It's only a few minutes in the wash, really.
Okay, could you set my flush, or sorry, my slurp to like 50, 60%, please? Actually, I'll leave this on. We'll flush the hose clean. Roger.
Let's see, where are we? How about this way, Emil? How's that look?
Okay, you can kill it now. That's right, yep.
I can't hear you, Emil. Is that good?
Yeah, it looks good.
Okay.
Let's try it.
All right, that's going to be, and then I think from there,
just straight on up.
Straight on up. I just want to get up to
Yeah.
this. Yeah,
sounds good.
It'll be slightly different, but for here we'll stay on the backscatter bit. I'm going 015.
Yep, good.
Bridge nav. The 40 meters bearing 015.
Oh, that looks
Set,
like
thank you.
urchin
Yeah,
test.
probably
Urchin.
an urchin.
We've seen quite a few urchins on this dive. All sorts of different flavors of urchin.
How about grape?
I haven't seen one of those. No.
Well, then I guess you haven't seen them all.
If you have time before that move kicks in, maybe you could back up and take a look at that urchin.
Sure.
It was kind of shiny.
We like shiny things.
There it is.
Oh.
Maybe not an urchin.
No, I think you're right.
Yep. All right. A crinoid. These are pretty yellow rocks.
What's that on the bottom right below the shrimp?
It might just be one of these yellow rocks.
No.
Or
Speaking of.
pinkish.
Yeah.
Okay, zoom in, please, Ed.
Wow, he's spiny.
Is that one a diadema genus?
Looks like potentially Sparosoma. These
Yeah.
white ones. It's hard to tell. This one's pure white. It doesn't have black on the test. No. Don't trust me on that one.
I'm not going to trust you.
Okay, there's a little
Never do.
crab or something under that rock.
It's definitely an Echinothuridae.
Do you want to see that yellow thing below us, or are we confident that's a rock?
Pretty sure it's like this thing just to the right. It's kind of oxidized.
Yeah. Okay, come wide, please.
Bye.
Yeah. All right.
Can you guys in the front row hear all the turns outside?
Not from in here, but we
experienced it, yeah.
We can hear him back here. It's a little strange.
Yeah.
You can hear him from inside.
I can hear him sitting here.
Wow.
Reminded me of
The
last
birds!
leg.
The birds! Oh, yeah.
When
Also
we had
that.
to dark out the boat.
Yeah.
That was lame. Those
It
poor
was
birds.
a kind of a tremendous sunset tonight. Boy.
And rainbow.
Yeah,
Yeah.
rainbow right over Baker Island. But the number of birds over the island was astounding.
Do we know what kind of birds are over the island?
Yeah, there's 11 species out there. I know they got brown boobies because we had one on board this morning.
Yeah, on the crane.
Watching the launch.
And I guess there are terns above us at the moment. Oh, we've also seen masked boobies hanging out near the bow.
Yeah, oh, cool. Terns, several species of terns, I think.
Left and right.
You know, oh, I was going to say to every season there's a tern.
Good, yeah, that's good.
I'm glad you didn't.
Trevor beat me to it. Bad joke.
I like yours better. Yeah.
So they're chirping out there?
Oh, man. Well, you just came
Guess you
in,
could
didn't
say
you?
that it's a turn signal.
Yep, there it is.
There it was. Heads in the background. No, no.
You know who hates bad jokes even more than you, Ed?
I'm
Is Wayne. Wayne hates them.
Oh, good.
Good.
Very good.
I'm just glad I'm on a watch with an Argus
Bridge
pilot
now.
I can depend on. Thank you.
One more step. Thank you.
There's some bird on the island, a type of noddy, a black noddy or brown noddy that the, an old whaling captain said they were an exceedingly stupid bird.
Exceedingly stupid.
I wonder what
Rather
experience the
rude.
captain had with the
That
bird.
mean.
It might mean
warrant
they were
that.
easy to catch. I don't know.
Yeah. Okay, so it seems like there's also frigate birds in Baker Island.
Oh yeah, frigates.
Bullies of the air.
They're gorgeous, though.
Naughties.
Great flyers.
Red-tailed tropic bird. Plovers. Now, does anyone know if those birds are migrating towards the island
Squishy.
and then leaving, or if
No,
they're
I-
residential
I
year
think they're
round?
resident, and they just forage for hundreds of miles.
That was actually a pretty noticeable thing for me on our way here towards Baker. We would see birds out of nowhere in the middle of the ocean. Like
Yeah,
the-
200 or 300 miles is kind of typical for some of these birds on the island. So they're a good indicator of a healthy ecosystem because they succeed if they've got small fish near the surface, and you have small fish near the surface, you have big fish underneath chasing them. So it's kind of an indicator that you've got tuna and other larger fish down there that are doing well.
I like when a shrimp goes right beside the light and it casts a huge shadow.
Yeah, like, oh no, and you look in Argus and it's just
Yeah, in
speck.
Argus view. What's this
What
little
is that?
black thing?
Ctenophore?
I don't know.
Oh, it's got little
Little wings.
Wings.
Oh,
This is gonna be cool.
I think
This
it might
is
be.
Go ahead and image it if you
It's
can, Ed.
flashing.
Oh, it's lasers.
Is it? No, that's
No,
it.
That's it.
That's it, yeah.
The
Really?
laser- yep,
Oh, it
there's the
is.
laser.
It's a
Lasers
It
are
is
Wow.
definitely a Ctenophore.
it's a Ctenophore.
Wow.
But it has some projection.
Whoa. Ah, where's he going? Oh, going back down. Ah,
Thruster.
I can't keep up.
Don't go.
He is really lighting up.
It really had
Wow.
its appendages really spread out.
Yeah, it was like gliding.
There's the gooseberries that do that.
This looks
That's
like a
quite
tooth.
large. It's about 10 centimeters, right? When we had the lasers on it.
Lasers are off now.
Well,
Roughly,
earlier,
yeah, across, yeah.
Okay, there's another one in the bottom left corner. Another one of those little
little sacks. Look at
Wow.
the colors.
thing that we this thing that we sampled is in the background.
Nice
Yeah, I see that
refraction there.
Man,
Awesome.
red, blue, green.
Is that what it is? It's refraction?
It's refraction. Yeah,
Yeah,
it's the same effect as a
refraction.
CD.
With its, was
glancing
it
light.
cilia or something?
Yep. Yeah, moving really rapidly up and
Yeah.
down.
Look
It
at
is.
it.
Oh, man, is it ever.
Yeah, the cilia are like paddles moving really fast up and down the body.
Such a small little critter with no real fins or anything. It still trucks along at a good clip.
I did not know they had those appendages. Boy.
Yeah, they'll extend them to fish, and
Huh.
each one will usually have even smaller tentacles drop off of it, like a fishing net.
Huh.
Come back.
Ah. It's a photo bomber.
It's
I'll say.
in the
Wait,
lens.
so do these type of ctenophores
Stop, get away.
have stinging cells?
They don't. They have they're kind of sticky tentacles. Wow!
Rainbow.
That's
wow.
awesome. It's like a party.
Yikes.
He keeps coming at me.
Yeah, I think he likes
Backing
it.
away as fast as I can, but he's chasing me.
He's an aggressive ctenophore.
Yeah, sure showing me who's
Go up. He
ruler of
probably
the
can't. It probably can't go up and down.
It's been going up and down like crazy.
Really?
Yeah,
Trained under Sodor.
I'll say.
Turn on the slurp.
Can't get away from it.
I can't. I've been backing up this whole time.
Yeah.
Can you use the slurp in reverse mode like a fire hose?
Just blasted away.
All right, I should move along.
Yeah.
Wow.
That was
nice
fun.
Spot.
That
Nice
was
spot.
cool. Yeah.
So, in answer to the question about stinging cells, the ctenophores like that
There's one.
instead have another one already.
Wow, he's still got his wings deployed.
We'll never get into waypoint 3.
We've been attacked.
We can do another move, Rennie.
Yeah. Bridge now.
Night of the ctenophore.
One more step.
So about the stinging cells.
Oh yeah. So the ones I'm familiar with are generally more in shallow water, and they're small. They're round, and they're usually called sea gooseberries, and they have a similar kind of fishing net tentacles with stinging, not stinging cells, but sticky cells called colloblasts. So they have like an adhesive quality instead of the stinging cells that you see on
Yeah.
the Nadaria jellies. But now I'm gonna try to figure out what that was. That was really cool.
Yeah, that's awesome.
It's not a body shape I've seen before down here.
Yeah, I'd like to know the name of those arms or wings or whatever they're called.
They were very graceful when they were extended. Fringes. Yeah. Stinging. Those are the cells. This.
Reminds me of like solar sails.
The fringes hanging down from the
Yeah,
tentacles
so
are the
yep,
stinging cells.
they have two
Sticky.
long, two long tentacles that shoot out, and then each tentacle drops like a fringe net
Yeah.
of smaller tentacles. They're so cool to see. Some aquariums will have them sometime. The smaller sea gooseberries, and you can spend hours watching them.
So you call those tentacles?
Yep, those would be tentacles.
Okay.
Yeah. And then colloblasts are the sticky cells instead of nematocysts, which are the stinging cells.
So that's how they trap their prey, correct?
Yep. Yeah. So they'll have just this fishing net that's see-through. Oh, is that a crab?
Yeah, we got a crab.
Where are we looking? Oh, crab, yeah.
Crab.
You wanted to image it?
Whoa. Oh,
I don't think it wants to
not
be
so
imaged.
fast.
See ya.
What's the spongy thing on above the lasers, though? Upper right.
Maybe it's a sponge.
Could be a
Could
sponge.
be. Could.
Go ahead and zoom.